Stroke is a brain disease that causes neuronal and functional loss. You can see the damaged portion of the brain in this image where it says infarct. Largely, over the time, some sort of recovery or compensation from the loss of function is seen. The extent of functional recovery is thought to depend on the ability of nearby brain regions taking over the functionality of lost brain region. Contemporary approaches are limited to exercise and training. Here, we are proposing a brain-computer interface for mice to map their brain activity with the sensory feedback. We can target the brain regions nearby stroke site and train the mice to increase the sensory feedback. We want to know if stroke-affected brain functions and brain areas can be brought back to action through this method. We also want to assess this system to determine some parameters for human BCI in the future. So this is my current work at Dr. Murphy's lab. My name is Pankaj, and I'm going to talk about real-time sensory feedback of cortical activity with a BCI. We are employing transgenic mice expressing GCAMP indicator cortex-wide. We are imaging the whole dorsal cortex at a mesoscale level. Here is a view of mouse whole brain in 3D by Murakami et al. 2018. We are imaging the dorsal projection of such brains. And this is the dorsal projection of the mouse brain that we image, overlaid with Allen Institute mouse atlas. Our BCI setup involves head-fixed mice. We use 470 and 440 nanometer wavelengths for optical imaging through a Raspberry Pi camera. Raw brain images, as you see in green, are processed in real time. You can see the processed activation map on the right side. We can choose target cortical regions R1 and R2, which are overlaid on the activation map. We continuously track the activations in these target regions, as you can see in this time trace. We can also impose arbitrary rules on these target activations. For example, R1 minus R2 will be mapped to the sensory output. This would automatically map R1 minus R2 activity to the sensory feedback, for example, audio tone feedback. So we are using an audio as a sensory feedback. In this figure, you can see the automatic mapping of R1 minus R2 activity with the frequency of audio feedback. We also set a pre-computed threshold above which the mice are rewarded with a drop of water. We expect water-restricted mice to learn to go above this threshold over the time, meaning they are to activate the target R1 and R2 regions based on the rule. The sensory feedback delay is typically around 100 milliseconds in our system. Here, I will show the system in action in this video. Left pane is the raw brain image. Middle is the processed activation map. And on the right, you can see R1 minus R2 activation in the red trace. Horizontal green line is the set threshold, and vertical blue line is the current time. I'm going to play the video briefly and hope you can hear the audio with that. So with this system, we did following experiment. We implanted cranial windows on mice on day 0 and let them recover for 7 days. And on the 7th day, we started habituating them and then move on to training in the closed loop system for 15 days. We trained several mice with different target regions and different rules. In this figure, each row represents reward-centered brain activation for different target regions and the associated rule during the training, as labeled on left of the image. We also found some evidence indicating the brain ROI activations change with the change in task rule. We did linear regression on the ROI DFF data and interestingly found that slope of the regression shifting towards the ROI in the rule. For example, here the left two plots are for the condition RY1 greater than RY2. And you can see the green regression line on day 3 compared to day 1 is more shifting towards RY1. On the contrary, in In the right two plots, when the task rule was switched to RY2 greater than RY1, in the same mouse, the slope is starting to shift towards RY2. With this system, we trained 19 mice with different target regions and rules over 15 days. We find that normalized number of rewards significantly increasing over the days. Further, we are trying to find and segment repeating brain patterns using an algorithm called seq-NMS over the training days to find brain patterns that could be task assistive in a rule-based manner. These motifs can be quantified and clustered into groups, as shown here. Each dot here represents one brain pattern that you see in the background. We are still working on ways to characterize and quantify them over the time. In conclusion, we present a real-time, low-cost GCAM-based optical BCI, where mice can be trained to modulate their brain activity in target brain regions. Using this, we can assess and probe different brain regions quickly and determine optimal parameters for BCI control. In future, we are trying to assess efficacy of this system for stroke recovery training. We are also integrating this BCI into the in-house home cages for long-term study. Additionally, we would like to test this system on different indicators as well as on multicolor expression in the brain. With that, I would like to thank everyone for their time and attention.